Hi guys, here's a little video to help walk you through the PowerPoint for 6.3. This PowerPoint deals with defense against infectious diseases. You can also think about this section of the textbook as discussing the immune system. So let's get started. The first couple slides are the understandings, applications, and skills. You can go through these on your own. They're also in the beginning of your packet. And hopefully we'll touch on most of these during the lecture. So the first thing we need to understand when we're talking about defense against infectious diseases is what exactly is a pathogen. And a pathogen is any organism or virus that can cause disease. So pathogens have antigens on their surface that your body recognizes as foreign. These antigens are usually like some kind of surface protein that your immune system will recognize and attack because your immune system can tell that it's something that's not supposed to be in your body. There are 5 major types of pathogens that we talk about in biology. There are pathogens that are present in bacteria, in fungus, in protozoa, in animals, and in viruses. An example of a bacterial pathogen is something like E. coli. I think that might be the one that's in this picture. No, it is not. But that's another example of a bacterial pathogen. A fungal pathogen is something like athlete's foot, and that is in this picture right there. A protozoal pathogen is like the pathogens that cause malaria or giardia. Protozoas are single-celled protists. They can be free-living or parasitic. In the case of the ones that cause disease, they're usually parasitic. Animals can be pathogens like ringworms, for example, or tapeworms. And viruses, of course, can be pathogens. That's everything from like the flu and a common cold to a virus like HIV. All right, let's talk a little bit about how we defend our body against all of this stuff that is trying to kill us. Thankfully, we have immunity, or the ability to resist infection by disease. And our immune system is what controls our immunity. There are 2 levels of immunity that people have. The first is non-specific or general immunity, and this is the ability to resist infection by a wide range of pathogens. So these are the parts of your immune system that just fight all pathogens, and they're not super specific about what they are. You also have specific or second level immunity, and that deals with the production of antibodies. And antibodies are like specialized proteins that fight specific antigens. You build up this specific immunity throughout your lifetime as you're attacked by different kinds of antigens and produce antibodies for them. And we'll talk about that a little bit later in the lecture. Let's start with your non-specific immune system. Your primary external defense in non-specific immunity is your skin. So your skin has 3 layers. The outer layer, the outermost layer, is called the epidermis, and this is mostly dead cells. So you can see the epidermis like right here in this picture, and this gives you a physical barrier against disease, which is great. Those dead cells provide like a layer of protection between you and the world. Below that, you have your dermal layer of skin, and this tissue is very much alive. In your dermal layer, you have sebaceous glands that secrete a fluid called sebum, which is like an oil that lowers skin pH to inhibit bacterial growth. So that is another way that that your skin tissue helps fight off disease and pathogens just in its very existence. Any place that you don't have skin, you need something else to help cover your immune system. So any place that you don't have skin, you have what is called a mucous membrane. And your mucous membranes produce mucus that helps trap pathogens. So these are present like in your nose, in your throat, in your vagina, in your urethra, any place where you have an opening where pathogens could try to get in. Um, and between those two, the skin and the mucous membranes, you have like a pretty good primary defense against pathogens that are trying to get into your body. Once a pathogen has like broken that initial barrier of skin or a mucous membrane, you meet the second part of your sort of generalized immune system, and this has to do with your blood. So we talked about blood in 6.2, and as a reminder, the major components of blood are plasma, red blood cells, which do not have so much to do with immunity, but they are part of your blood, and platelets and white blood cells, and these are super important in your immunity. So like, let's see, platelets here are little cell fragments, and we'll talk about those because when you get a cut, platelets are super important. Then you also have all different kinds of white blood cells present in your blood, and I'm circling some of them on this picture now. They deal with all different kinds of immune response in your body. So those are some of the examples of types of white blood cells that are present in your blood. You don't need to memorize any of those names except for a macrophage, and we'll talk about those in a little bit. All right, so first thing, if you have a cut in your skin, that is a super easy way for pathogens to start to invade your bloodstream. So your body actually has a pretty specific response to deal with if your skin gets cut. Your platelets in your blood rush to the site of the cut and form a sort of temporary plug. So you can see like this is supposed to be a break in your skin. Platelets are rushing to the site of the infection. Those platelets release clotting factors that produce an enzyme called thrombin. And thrombin is super important because it allows for the catalyzation of fibrinogen into fibrin. So you basically have this soluble type of a protein present in your body. When thrombin acts on that protein, it creates an insoluble protein that allows you to sort of, what's the word I'm looking for? It allows you to like create a layer of cells. It acts as like the glue in a layer of cells over that cut that plugs up wherever your skin is broken. So that fibrin mesh catches all of the other stuff in your blood, red blood cells and platelets. You can see them all like gathering up here. And that is what forms a gel. And you'll notice that, like if you have a cut in your skin, there's sort of like a gooey layer that forms over it. And that gel is eventually what hardens into a scab and works to protect the area of your skin that's been like breached basically by this cut. So the next slide, there's a little video of this happening in real time of like what happens when a cut is sealed up by a blood clot. And you also have the two benefits of the ability of your body to do this. The first is obviously that it stops bleeding. Like when people are hemophiliacs, they're missing blood clotting proteins and they're not able to stop bleeding, and it can be super dangerous to get a cut. It also obviously prevents pathogens from entering your body, which is great because that is like the ideal way to deal with pathogens is to just not have them enter your body in the first place. However, this idea of blood clotting can go slightly awry sometimes. And if you have a blood clot form in a place that it is not supposed to because of these clotting proteins, it can actually cause some real problems with oxygen circulation in your body. One place that we especially see this is in your coronary arteries. We call that coronary thrombosis. A thrombos is a blood clot. So when you have blood clots forming in your coronary arteries, we call that coronary thrombosis. As a reminder, your coronary arteries branch off from your aorta to supply oxygen to the heart. So like they go right back around from the aorta into the heart to give that cardiac muscle the oxygen that it needs to keep functioning. And if that muscle doesn't get the oxygen that it needs, that's a big deal, because when cardiac muscle is deprived of oxygen, the muscle tissue starts to die off, and that muscle tissue cannot be regenerated. So if you have coronary thrombosis, usually caused by like smoking or diabetes or obesity, you can have a blood clot form, and that limits the oxygen flow and nutrient flow that is getting to that heart muscle. So when you have that limited oxygen and nutrient flow, obviously you're not passing on glucose for cell respiration, you don't have enough ATP, and your cells are not able to function regularly. This leads to irregular heartbeat and eventually can lead to death. This blockage in your coronary arteries is like what we sometimes refer to as a heart attack. And those are bad news. But getting back to the immune system, let's talk about what happens if you have a pathogen that is able to pass through your primary lines of defense, your blood and your skin, into your bloodstream. We still have some primary immune response available to us. Like, we don't have to have a specific specialized immune response for every single pathogen that gets into our body, which is great because it actually like takes a fair amount of energy for our bodies to have that specialized immune response. So our primary immune response in our bloodstream is a type of a white blood cell called a phagocyte. And specifically, the phagocytes that we look at in this class are macrophages. These are white blood cells that eat pathogens sort of indiscriminately, like anything that a macrophage recognizes as not being from your body. It will just gobble it up through endocytosis, and they're packed with lysosomes. So they'll take in whatever, like say these in this picture are virus cells, they'll take them in through endocytosis and then digest them using all the lysosomes that are present in the cell. Like you can see these lysosomes are coming to get this virus cell, they digest it, and then produce this waste material that's then ejected from the cell. Pus is a byproduct of this specific chain of events. So macrophages are great because they give us like just some more general immune response. There's another picture of that process happening where a microbe or like a virus cell is adhering to the phagocyte, usually a macrophage, and then the offending particle is getting broken down. That's like just another version of that happening is in this picture. As all of this is happening in your body, you have some reactions to this. The first big reaction you have is inflammation. So when white blood cells rush to the site of some kind of an injury in your skin, that causes inflammation. And the thing is, your body doesn't necessarily know like what is happening when you have an injury in your skin. It could be just like like something got into your skin. It could be like an invasion by microbes, but your body does know that white blood cells need to rush to that area. So they increase dilation to allow white blood cells to access the damaged tissue. And as all of those white blood cells rush into that area, you see redness, heat, and swelling, like localized redness, heat, and swelling in the area where you're dealing with some kind of a breach to your primary immune response. You can also develop through your primary immune response a low-grade fever. And that is because if your body is sort of fighting something off, it's going to increase the body temperature. This is helpful for a couple of reasons. It inhibits the growth of microbes by denaturing their enzymes, and it also facilitates that process of phagocytosis. It allows macrophages to move around your body more quickly and gobble up all of the offending pathogen molecules. So that's why you get a little bit of inflammation and a little bit of fever when you're fighting off some kind of a pathogen. If things get more serious than generalized immune response, we move into specific immune response. We also call this acquired immunity because in your specific immune response, you have the development of antibodies that you acquire, like sort of over the process of your life. So when we look at specific immune response, we need to understand how to define a few things, first of all. And one very important term is antibodies. So antibodies are large proteins produced by lymphocytes. Lymphocytes are a type of white blood cell. And they are specific to certain antigens. So you have a specific antibody that bonds with every specific antigen that you have ever had an acquired immune response to. There's a region that binds to the specific antigen, and you can see that on this picture here. Like this antibody has a region that binds to the antigen right there. And the reason that that happens is the antibody is able to sort of mark the antigen for destruction in that way. And then there's also a region that prevents the viruses or pathogens from attaching to the host cell or makes the pathogen recognizable to phagocytes. So that's like the idea that I was just talking about, where this antibody acts as like a flag to your immune system that something's going wrong and something needs to come and kill this antigen. Kill the antigen. Blah. Die. All right. Let's, now that we've brought up this idea of lymphocytes, define them a little bit as well. So lymphocytes are a class of white blood cells that include T cells and B cells, and we are going to talk a lot more about T cells and B cells and their role in your immune system right now. So the generalized function for two different types of T cells and for B cells is present on this slide. Take a minute to pause the video and write out in your note packet the function of killer T cells, helper T cells, which are these ones, and B cells, which are these ones. And then we will go over how those cells interact to allow your immune system to kill off pathogens. So there's 6 steps in this process, but I sort of compressed it down into 5 on this slide because 5 and 6 are sort of similar. So the first thing that happens when you have a specific immune response is the pathogen gets ingested by a macrophage, and their antibodies get displayed in the plasma membrane of the macrophage. So you can see that happening here, where the macrophage ingests the pathogen and then displays its antigens. That should say antigens. Here, this should say antigens. All right, and then once those antigens are on display, lymphocytes, remember lymphocytes are white blood cells called helper T cells, arrive and help to figure out what the correct antibody for the situation is. So helper T cells specific to that one antigen are activated by the macrophage. And those activated helper T cells bind to a different kind of lymphocyte, a different kind of white blood cell called a B cell. And the B cell is the kind of cell that sends out the signal to manufacture a ton of whatever the right kind of antibody is. It's super important that we get a lot of antibodies manufactured because we want those antibodies to go out and mark as many of the offending pathogen cells as they can, as quickly as they can. So plasma cells are the B cells that are responsible for that. They produce and secrete a large number of antibodies during an immune response. Let's see, you can see that happening as we go through the other steps of this picture. So a B cell specific to the antigen is activated by proteins from the helper T cells. The B cell divides repeatedly through mitosis to produce antibody-secreting plasma cells. That's what we're talking about down here, are those plasma cells that are going to make a ton of whatever the right kind of antibody is. And the B cell also divides to produce memory cells. So you have two things going on here. The first is the immediate response, where you're like pumping out antibodies as fast as you can. That doesn't last for so long because once you've taken over the antigen and overpowered it with your immune system, those antibodies go away. So your immune system also produces these memory cells that allow you to have, the next time that you come in contact with that specific type of an antigen, you have a little bit of a memory of how to make the correct antibody, and that allows you to fight off an infection faster the second time that you get it, which is great. So that is the general path of specific immune response. Let's talk a little bit now about human immunodeficiency virus, or HIV, and its effect on the immune system and its methods of transmission. So HIV is a virus. It's from a family of viruses called retroviruses. And the bummer thing about this virus is that it attacks your immune system. It attacks your lymphocytes, your white blood cells. So obviously, when these cells are under attack, eventually they lose the ability to make antibodies. So you lose specific immune response capability. Once you've lost enough of your specific immune response capability, like once your body has lost the ability to produce a certain number of antibodies, HIV virus leads to a syndrome called acquired immune deficiency syndrome, and we call that AIDS. At this point, an individual's immune system is compromised enough that they can really no longer fight off pathogens. So when people have AIDS, they tend to die of some kind of secondary infection, like maybe you get a cold or the flu, and your body really just isn't equipped to do anything to fight it off. So since your immune system isn't functioning properly, those pathogens take over your body and they kill you. And that's why HIV and AIDS are so serious. Transmission of HIV is through bodily fluids. This can be like sharing needles or unprotected sex, or occasionally a mother can pass HIV positive mother can pass HIV on to a child during childbirth. It's not guaranteed that an HIV positive mother would give birth to an HIV positive child, but that it can happen during the process of childbirth. And let's see. Next, we're going to talk a little bit about how we fight off some kinds of pathogens. And this has to do with Alexander Fleming and the discovery of penicillin. So Alexander Fleming was a scientist in 1928 who discovered this mold substance that contained what we now call an antibiotic called penicillin. And penicillin, the discovery of penicillin was accidental. Basically what happened was Fleming left out a dish containing Staphylococcus aureus bacteria, And when he went back to check on the dishes in his lab, he noticed that this mold had started to grow, and that where the mold was growing, the bacteria was not growing. So you can see like the original experiment from Fleming here. This is a penicillin colony, and then this is where it's sort of killing off the bacteria, and this is the normal bacterial colony over here. We can still do experiments like this, and a modern experiment example would be more here, like you have a penicillin colony up here, and then a streak culture on an agar plate, and you can see in this area that streak culture of bacteria isn't growing, and that's because it's reacting to the penicillin that is on the dish, and it can't get too close to the penicillin because the penicillin is killing it off. So the discovery of penicillin was a big deal, but Alexander Fleming never quite nailed down how to isolate the compound correctly in order to use it in medicine. That part of the story involves two other scientists called Howard Florey and Ernest Chain, working in Australia in 1940. So these scientists tested penicillin on mice infected with a form of streptococcus. And they noticed that the mices that were injected with doses of isolated penicillin were okay, and the untreated mice died of the bacterial infection caused by streptococcus bacteria. So in this way, we realized that this substance could be considered an antibiotic and could be administered to other living organisms in order to fight off bacterial infection. This was a big deal, and in 1945, the three scientists together were awarded the Nobel Prize in Medicine. The problem that we have now with antibiotics for treating bacterial infections is we use a lot of them. So antibiotics can only be used to treat bacterial infections. Remember, bacteria are prokaryotic cells. Some differences between prokaryotic bacteria and eukaryotic animal cells can be found here. But basically, the majority of antibiotics work by attacking peptidoglycan in bacterial cell walls. Eukaryotic animal cells don't have cell walls, so that means that antibiotics can kill bacteria without harming the host. That is also why antibiotics don't work on viruses. The problem with using a lot of antibiotics is due to genetic variation, there are often some members of a bacterial colony that can resist antibiotics. And as you use more and more antibiotics, it's more and more likely that those resistant bacteria are going to be left over from a population, and you're going to need stronger and stronger antibiotics to try to kill off the bacteria. One really good example of this is called Mrsa, and that stands for methicillin-resistant Staphylococcus aureus. And this is getting to be a real problem, like in hospital environments, because this is a very powerful strain of bacteria that is very resistant to most antibiotics. So when people come down with a Mrsa infection, it's not unusual that that Mrsa infection can kill them. We talked about this a little bit in 5.2, like some ways to really try to work against bacterial resistance are to make sure that you take the full course of antibiotics anytime that you're prescribed antibiotics, so that those antibiotics kill off as many of the members of the bacterial colony as possible. All right, and that about does it for the lecture for 6.3. Next class, you guys will have a formative quiz with the mark scheme so that you can grade yourself. There's also at the end of this PowerPoint another video if you would like to have just like a little bit more understanding of the specifically really like T cells and B cells are explained pretty well in this video. There's also an alternate down in the notes if you don't like this video. All right, and that about does it, and I hope that this was helpful. See you soon.